Don't you just love that cartoon? I found that on Eddie D. Robertus's website, which looks like it might have been made in 1995, but that graphic was pretty cool. At any rate, in this episode, we are going to be learning about some of the molecular things that are happening in the organizer and what they are doing. So we're going to start off with another interesting rescue experiment. This time it's from Gerhardt. We're also going to look at the SMO in amphibia and realize that it's actually the same thing as the shield in zebrafish. We'll be looking at the molecular signaling in the organizer. Talk a little bit about dorsalizing versus ventralizing factors and how that works. We'll end up discussing what's now known as the double gradient model. And we'll see how TGF-beta proteins and BMP and Wnt gradients integrate both the DV axis and the AP axis. And then finally, we'll talk a little bit about regionalization of the embryo in the setup for post-gastrulation development, and a little bit about evolution. All right, let's get started. Okay, so we're looking at a slide here that depicts an interesting experiment that's not that different than the ones that you saw from Spemann, except this one was done in the 1980s by Gerhardt, and he's the gentleman that did the gravity rescue of the UV single-cell embryo that we looked at earlier. So what Gerhardt's doing in this situation is we've got two fertilized eggs. One of them gets exposed to ultraviolet light, and you know what happens. You don't get tubulin polymerization, so you don't get the tubulin microtubular network at the shear zone. You get no cortical rotation, and you get the Baustock, the belly piece, instead of a normal embryo. So what he showed was you could actually transplant a cell from, we're talking here like a 16-cell embryo. You could take a cell that's in the region that will become the dorsal blastopore lip and transplant that to the UV exposed embryo and rescue it. If you don't do the transplant, no rescue, okay? And if you do the same thing without even bothering with the UV, if you just use a normal recipient and a normal donor, you get a new gastrulation site and a second body axis, just like you did with the newts in Spemann and Mangold's experiment. Okay, now let's change gears a little bit. What you're looking at on the screen, this art is right from your textbook. This is, on the left, our xenopus embryo, and on the right, we're going to be contrasting it with the zebrafish. So we've been focusing on the amphibian. Just going to remind you that the zebrafish has a very different looking blastula and gastrula. We've got that cap sitting on top of the yolk in the fish. Okay, so this is showing you some of the same things. You're already familiar with all this. I just want you to see how it works in the fish. So we've got those dorsal determinants that are basically disheveled during cortical rotation, moving up dorsally, and we've got the beta-catenin that is enabled to go into the nuclei of those cells in the dorsal region due to that stabilization. Same thing is happening in the fish, all right? And what you'll notice now is that the region that is doing the signaling in the zebrafish is basically in the same location, but it's a little bit different, and we call it the shield, not the organizer or the dorsal blastopore lip, because there is no dorsal blastopore lip. The shield is the organizer in the zebrafish. Okay, now we're going to change gears one more time here, several more times actually, but what I want to mention to you next, just take a step back and put some of this in context. We've been talking about early embryology and really the transition between the fertilized egg, the evolution into the blastula, and the transition into the gastrula. All right, and we've talked about the idea that early on it's all maternal effects, and that the zygotic genome is not even active. And I have mentioned to you in class before this idea of the mid-blastula transition, which is that time when the zygotic genome gets turned on. There's another name for this. So we've got the mid-blastula transition, the MBT. We also have another term called the mid-zygotic transition. All right, and they're not exactly synonymous, but for our purposes, we'll just treat them like they're essentially the same thing. And what I want to point out here is that all of the events that we are talking about are occurring during this time. So before the zygotic genome is completely turned on, we've got a situation where we have rapid cell cycles with synchronous divisions. That means that every cell divides at the same time. So when you're looking at an 8-cell embryo and it goes into that 16-cell embryo, it's all 8 of those cells are dividing essentially at the same time. Okay, so the first cell into 2 cells, that's synchronous. Those 2 cells into 4 cells, generally synchronous, etc., etc. And rapid cell division, okay, relatively rapid cell division. Once you start turning on the zygotic genome, we start to see some other things happen as well. We start to see asynchronous cell division, slower cell cycles, and an increase in cell numbers. We also see a decline in the initiation factors that were provided by, I should really just say the transcription factors that were provided by the mother to turn on those zygotic transcriptions. All right, so another way of looking at that is here. This is using a zebrafish as an example, showing you that time before the MBT, and then at that mid-zygotic transition point, we start to see increased cell cycle length, introduction of the G phase, and an increased S phase time. All right, we'll leave the EGT and the ability to perform apoptosis for another time. Just want to set that into context, set all of this into context, and return to our topic at hand, which is the molecular activity at the organizer. Okay, now let's return to the molecular organization that I wanted to talk about in this podcast. So this cartoon should be easily interpreted. It's a late blastula. The organizer has been induced by the nucleus center. What's doing that induction? Well, it's beta-catenin. All right, so you basically have these mesodermal inducing signals and endoderm inducing signals, which are maternal effects that are coming up out of that vaginal pole, coupled with the cortical rotation and our stabilization of beta-catenin. We then change that dorsal aspect from the rest of the embryo. All right, so you've seen this slide before. I'm just using it here to point out the factors that are involved and what the players are. VEG-T, VG1, these are maternal effect messenger RNAs. The VEG-T is endoderm specifying. VG1 is a mesoderm specifier. And coupled with the fact that VEG-T also induces XNRs, nodal related proteins, to be produced, we get that nucleus center turned on, which induces the organizer just above it at the prospective blastopore lip. Now, what else is happening is we've got mesodermal signals that are starting to spread through that presumptive mesoderm, and we'll talk about what those are in just a moment. Just quickly illustrate again that that dorsalization signal is really the result of beta-catenin and the Wnt pathway. All right, so I'm just emphasizing that to you now. All right, and it's coupled to the TGF beta signals because we've got SMADs that are involved in this, but that's a little more than I want to get into at this level. All right, so what else is going on there besides beta-catenin? Well, we've got those TGF beta signals, and the big one that you're already familiar with is BMP. Now, there's probably a dozen different BMP molecules that have been identified and numbered, certainly 9 or 10, but I'm not going to ask you to remember that it's BMP4 or BMP3 or BMP2. Just remember that it's BMP. All right, so BMP, which is a member of the TGF beta superfamily, is the mesodermal signal that is, it's actually a ventralizing signal. All right, so it's not just mesoderm, it's being expressed in that mesoderm, but it is a ventralizer. All right, so what do you think happens? Well, I mean, in other words, what do you think the organizer is doing? If you've got this ventralizing signal, this BMP, coming across that mesoderm, well, it makes sense after the fact. I mean, it took 20 years for people like de Robertis to figure this out, but in effect what's happening is the organizer is inhibiting the ventralizing signals. So dorsalization, while we can legitimately say that it's initiated by that cortical rotation and the ultimate stabilization of beta-catenin, what's really going to happen here, the downstream consequences of that, are the production of these molecules in the organizer region that are inhibitors of the TGF beta proteins, the BMPs. So BMP is default ventral signal. Inhibit BMP, and now you have an opportunity for dorsalization to occur. I'm probably going to say that 5 times in 5 different ways because it's that important for you to understand. So repression of the TGF beta superfamily proteins is what helps set up a concentration gradient. You've got BMP being expressed in a rather uniform distribution all the way across that mesoderm. It's being repressed. It's not being allowed to bind to its receptors by these things that are being produced in the organizer. So probably the most important one is the one that I had you read a paper about, which is cordon. All right, cordon is the major competitive binder of the BMP molecules, and by competitively competing with the receptors, it reduces or represses the signal. So you've got BMP being expressed in that mesoderm in a relatively straightforward manner, not much of a concentration gradient going on here. But in the dorsalmost region, you've got inhibitors, competitors to the receptor that are, they're not competitors to the receptor, they are proteins that bind to the BMP signal. So cordon binds to BMP. Matter of fact, I believe they were precipitated together when they were first discovered, because cordon has a high binding affinity for TGF beta proteins. So what's happening is you've got these TGF beta proteins being expressed, and then you've got cordon, which is tying them up and preventing them from binding to their receptors. All right. What else is really cool about this is that they're secreted and they are extracellular. So the morphogen gradients that we've looked at in detail prior to this, primarily in the fly, were things like bicoid that could diffuse through the syncytium, and you had a concentration gradient that was basically due to Brownian motion source to sink. You know, you could explain that almost entirely with thermodynamics and diffusion. This is similar to that, but it's happening outside of the cell. It's in the extracellular matrix. And that's a really, really exciting idea, okay, for a whole bunch of reasons, only some of which I'll be able to explain to you today. So if we look at, this is a fluorescence micrograph showing a section through through a frog embryo, and what you're looking at here is, if you look at that white line that is demarcated by the arrowheads on the left panel, that white line is cordon. All right, so cordon is being expressed it's being secreted. It occupies this very narrow little region called Brachet's cleft, which is a little virtual space in between two layers of cells in an extracellular acellular matrix, proteoglycans, collagens, things like that. And there's a definite concentration gradient where it's highest in the dorsal region because it's being expressed near the dorsal blastopore lip in the organizer region, and then it's diffusing out in that extracellular matrix. Now on the right, you've got a similar setup, but here they're labeling fibronectin, which is ubiquitous. So in Brachet's cleft, you see fibronectin, and it circumnavigates that entire embryo. So that white line goes all the way around. And this is basically just showing you that there's a very big difference between the way fibronectin is secreted. Fibronectin is one of those extracellular matrix molecules, and the way the cordon protein is secreted. There's a definite concentration gradient in cordon. So you've got that cordon being secreted, you've got the BMPs being secreted, and you've got cordon binding to the BMPs and then essentially preventing the BMPs from binding to their receptors. And that's establishing a dorsal ventral axis. Okay, that is the repression or inhibition of BMP is the dorsalizing signal, and the expression and reception of the BMP signal is the ventralizing signal. So the default is going to be, the default without having BMP bind to its receptor is going to be dorsal identity. When BMP is allowed to bind to its receptor, you get ventral identity. And it's that concentration of cordon, which is a BMP inhibitor, that prevents that from happening. Now, it's not the only BMP inhibitor. There's, a couple of other ones that are involved in this too, but it's certainly the major player. All right, now in the next slide, this is also a very familiar slide because it's showing things that we've seen already. This is the late blastula. This is the, it's a fairly late blastula in the first and second images there. And then we've got an early gastrula in the third image. And showing you the major players there, VEG-T, VEG-1, VEG-T being the endoderm signal, VG1 being the mesodermal signal, and then beta-catenin being concentrated in the dorsal region, and VEG-T turning on nodal. All right. Now, if you go below, we'll skip to the lower part of the diagram. We'll come back to that third embryo image in a moment. If you look at what's happening below, this is not a hard diagram to parse, but you probably need a little help. So what this is saying is that in the presence of beta-catenin and the presence of VEG-T and VG1, if you have a high dose of nodal, you're going to get dorsal mesoderm. And dorsal mesoderm is a synonym for the organizer. If you have a low concentration of nodal related and VG1 and VEG-T but no beta-catenin, then you're going to get ventral mesoderm. All right, and the ventral mesoderm is what's going to express X-Wnt8 and BMP4. All right, the organizer region is what's going to express the inhibitors of those TGF-beta molecules, and that's how we're going to ultimately establish the dorsal ventral signaling. Here's another version of the same information. Beta-catenin with high nodal, VG1 and VEG-T with low nodal, ventral mesoderm versus dorsal mesoderm. All right, so let's summarize what we're talking about here with the organizer in the following manner. While the new coop cells in the nucleus center are actually endodermal cells. What they are going to induce is the formation of dorsal mesoderm, and that's the organizer. All right. The organizer self-differentiates into this dorsal mesoderm. The dorsal mesoderm is producing signals that dorsalize the surrounding tissues. All right. And what you'll see is going to be very important after the next exam when we start talking about neurulation and things like that, is that the dorsal mesoderm induces neuroectoderm right above it, and it also is orchestrating the gastrulation movements that we see that everybody associates as being synonymous with gastrulation. Now, it's become fashionable recently to look at the late blastula and the early gastrula as a system that has two signaling centers, whereas traditionally we've thought of just the major signaling center in the stage 8, 9, 10 embryo as the Spemann-Mangold organizer, the SMO. Lately in the literature, we've been seeing this idea of two signaling centers, a ventral signaling center and a dorsal signaling center. The dorsal signaling center is the Spemann organizer, and the ventral signaling center doesn't have an eponymous name, but it's the region in the ventralmost pole where we're getting those TGF-beta superfamily proteins like the BMPs and things like that being produced. Now, I'm not going to make a big deal about this. I just wanted to mention this to you. The reason I'm not going to make a big deal about it is because there is a more important idea called the double signaling gradient model, or the double gradient model, which is too easily confused with this. So be familiar with this. For those of you that are interested in this kind of stuff and may go forward in development, be aware of this term. But what I really want to focus on is the idea of the double gradient model, and they're not to be confused. All right. The two signaling centers is the source, the ventral source of BMPs versus the dorsal source of BMP inhibitors. The double gradient model has to do with Wnt and BMPs. All right, so we're going to focus on the double gradient model. I can guarantee you I'm not going to ask you a test question on the two signaling center idea. I'm just giving that to you because I think it's kind of interesting. All right, so the double gradient model is this idea that axis formation, both DV and AP actually, is controlled basically by two signaling molecules. BMP we've already talked about, and Wnt. All right, and this picture that you see here, I'm going to show this to you several times in several different ways. This is the money slide, okay? So get that picture into your head. The green arrows are showing a gradient of BMP. The blue arrow is showing a gradient of Wnt. All right, Wnt is dorsal and is in a gradient from posterior to anterior. So the highest concentration of Wnt is in the posterior region. The lowest concentration of Wnt is going to be in the anterior region. BMP is ventral and uniform along the AP axis. It's not uniform as it is going dorsally from ventral. So along the DV axis, its highest concentration is ventral and its lowest concentration is dorsal. But along the AP axis, it's fairly uniform. And these gradients are perpendicular to each other. Okay, now this is going to be very important. Here's another illustration that doesn't show exactly the same thing, but gives you some of the same information. All right. So another important thing to get your head around here is that the gradients are established here in two fashions. They are established both by source to sink diffusion, and as I've already told you with cordon and BMP, they are established via active antagonism. So these molecules like cordon that prevent or inhibit BMP signaling are also in effect contributing to the overall gradient. All right, so the embryo as a whole has this rather uniform BMP4 signal, and it's got a high concentration of BMP antagonists in the Spemann-Mangold organizer. Okay, and then you've got that mesodermal gradient, of BMP, and again, that's being inhibited by molecules that are produced in the organizer. All right, just so you have a list, here are some of the proteins that are being expressed in the organizer, both in terms of nuclear transcription factors and in terms of secreted proteins which will bind to receptors. Not going to make a big deal. I'm not going to ask you to write a list of these things, but I may ask you to be familiar with some of these, okay, in the sense that they are organizer proteins. All right. Now, if you look at the combinatorial activity of Wnt and BMP along the AP and the DV axis, what you see is that varying concentrations of these two signals will result in different regional specificity in the ectoderm. All right, so these are signals that are being produced in the mesoderm that are acting in mesoderm and are also acting in endoderm and ectoderm. All right, so this is showing you, this particular slide is showing you what's happening in the pharyngeal endoderm region and in the mesoderm in the head and in the notochord. All right, so you can see that those signaling molecules that are inhibiting Wnt's and those signaling molecules that are inhibiting BMPs. Another interesting way to look at this, same type of information. This is just looking at ectoderm. All right. And what you can see here, and by the way, please note that this diagram is 180 degrees opposite of the first diagram. So posteriors on the left here, anteriors on the right. It was reversed in the other diagram. But this is showing you something very interesting. If you have the presence of both BMP and Wnt, that signal is epidermis. If you have neither BMP nor Wnt, you're going to get neuroectoderm in the head region. All right, so that's going to be brain. If you have Wnt but no BMP, you're going to get postcranial neuroectoderm. All right, so just those two signals, by varying whether they're on or off, Wnt and BMP on, that's a signal to make skin, non-neuroectoderm. Having no BMP and no Wnt, that's a signal for forming central nervous system, cephalized central nervous system. Having Wnt but no BMP, that gives you trunk and spinal cord. All right. So again, here's that diagram showing you the Wnt gradient and the BMP gradient. Remember that the BMP gradient is uniform along the AP axis, but non-uniform in the DV axis. It's higher concentration ventral, lower concentration dorsal. And the Wnt gradient is high concentration posterior, low concentration anterior. Another way of looking at this is looking at how Wnt and BMP are involved in intensity and duration of the signal. All right, so increasing duration and increasing intensity of the signal, Wnt and BMP, is really imposing a Cartesian coordinate system on this three-dimensional embryo. All right, another illustration showing something very similar. And also, I want you to realize that because we have this concentration, this double concentration gradient, we are actually establishing both DV and AP with the same two signaling molecules. All right, I know this is a lot of information and you're starting to get saturated. Just bear with me. I've only got one or two more really short but interesting things to tell you. The last things I want to talk about in this particular podcast have to do with some of the much bigger picture ways of thinking about these signaling systems. So on the slide that you are looking at now, What you see on the left is a vertebrate, and on the right is a fly. All right. And one of the things that I mentioned to you in the introduction to the course way back in January is that the vertebrate body plan is in many ways an inversion of the invertebrate body plan. All right. So we have a dorsal hollow nerve cord in vertebrates. We have a ventral nerve cord in invertebrates, just for example. All right. So one's dorsal, the other's ventral. Well, if you look at the signaling, the signals are the same, but they're flipped. Now, that's probably not surprising if you think about it. I just want to make sure that you think about it. So that's what you're seeing here. The dorsalization signals and the ventralization signals are flipped in the vertebrate relative to the invertebrate, and thus the ventral structures are flipped and the dorsal structures are flipped. All right? Now, along the same lines, you can do some drastic alterations to a chordate body plan by altering the size of the organizer region. All right, so what you're looking at here is using Xenopus as a model, changing the amount of organizer tissue present and the location of that organizer tissue. And what you see here really is a spectrum of results. And the spectrum is a dorsal, ventral, anterior, posterior spectrum where you've got ventro-posteriorization on one end of the spectrum and dorso-anteriorization on the other side of the spectrum. Now, why am I showing you this? Well, think about this. If you want to change a body plan, there are a couple of really simple things that you can do to make some drastic changes. Just altering where and how much of an organizer is present can completely and radically alter a body plan, just like flipping signals can alter a body plan. All right, just some big food for thought when you're looking at the phylogenetic tree and you're wondering how things happen. Well, interesting stuff. Okay, let's wrap it up. Okay, let's summarize. Well, we learned today that the organizer is called the shield in fish. We have reiterated that the organizer is induced by Newcoop Center. We've talked about the molecules involved. We discussed how the organizer produces antagonists or inhibitors of TGF-beta superfamily proteins. And we learned, interestingly, that the DV axis is essentially controlled by the BMP gradient, and the AP axis is essentially controlled by a Wnt gradient. And that the organizer, what we didn't talk about was things like follistatin that are inhibitors of Wnt. We talked about things like cordon that are inhibitors of BMP. But the fact is that the organizer is producing inhibitors for both of those kinds of molecules, and that it's integrating both axes via that double gradient model. We learned that cordon is the major player in BMP antagonism, and I gave you a paper to read about that as well. Interestingly, we learned that cordon is an ECM morphogen, which I think is really, really cool. And then I showed you some interesting evolutionary ideas where that flipping of the body plan from invertebrate to vertebrate is easily explainable by looking at the signaling molecules, which are also flipped. And then we looked at how altering the expression of some of the molecules in the organizer region by increasing and moving that region, we can drastically alter the body plan along a spectrum. All right, that's it for today. Thanks so much for your attention. This was a long podcast with a lot of dense information in it. I'll see you next time.